Hello everyone. Today we'll be solving Cambridge International AS and A-Level Biology Paper 4 A-Level Structured Question May-June 2021 Paper 4-2. We'll be solving today from question number 6 to question number 10. Question number 6. Glucagon-like peptide 1 GLP-1 is a hormone consisting of 31 amino acids. It is secreted by specialized intestinal epithelial cells known as L cells. Figure 6.1 outlines the secretion of GLP-1 and its effects on the pancreas, brain, and stomach. So we can see GLP-1, which is secreted. It goes to pancreas. In the pancreas, it increases in insulin secretion from beta cells and decrease in glucagon secretion from alpha cells. Alright, GLP-1, which is secreted, it goes to the brain. Then the brain decreases appetite. GLP-1 secreted, which causes inhibition. Alright, so it causes inhibition. The stomach empties, nutrients stimulate L cells in intestine, and as a result GLP-1 is released. Okay, so GLP-1 binds to receptor on cell surface membrane of beta cells in the pancreas. These cells secrete insulin. Name the type of membrane component that forms a receptor in the cell surface membrane. We know that glycoprotein and glycolipid forms receptors, so it will be either glycoprotein or it's going to be glycolipid. State how GLP-1 is transported to the pancreas. Well, GLP-1 is released in blood, so will be transported through the blood. So blood. Part B. GLP-1 inhibits the secretion of glucagon by alpha cells in the pancreas. Describe two processes in the liver that are stimulated by glucagon. We know that glucagon stimulates the breakdown of glycogen into glucose, which is basically known as glycogenolysis. Gluconeogenesis is also stimulated. Glucose is converted from fatty acid or amino acid. Part C. Suggest one reason why GLP-1 causes a decrease in appetite. Alright. So basically GLP-1 reduces ingestion of food. Part D. GLP-1 inhibits the emptying of stomach. This is an example of negative feedback. Explain what is meant by negative feedback. So basically a negative feedback is when there is a change in parameter, it is detected by receptors, and as a result a corrective action is taken by effector. The parameter then returns to the set point. Part E. A transcription factor is involved in the production of GLP-1. An insertion mutation in the gene that codes for the transcription factor can affect the production of GLP-1. Insertion effect outline the effect of an insertion mutation on the production of GLP-1. So insertion mutation leads to a frame shift. As a result, a different primary structure of polypeptide is formed. Transcription factor is likely to be non-functional. Gene coding for GLP-1 is not expressed anymore. Question number 7a. The fruit fly Drosophila melanogaster feeds on sugar found in damaged fruits. A fruit fly with normal features is described as a wild type. It has red eyes and its wings are longer than its abdomen. There are mutant variants such as purple eyes or short vestigial wings. Figure 7.1 shows a wild type fruit fly and mutant fruit fly with purple eyes and vestigial wings. So we can see the wild type, red eyes, long wings, and mutant one which has purple eyes and vestigial wings. Okay. The gene coding for the eye color and wing length are located on the same chromosome. So they are autosomally linked.
Autosomally linked.
Allele capital R for red eyes is dominant to the small r, which is purple eyes, and allele capital N for long wings is dominant to small n, vestigial wings. Explain what is meant by the terms allele and dominant. So allele is basically different form of the same gene, and the term dominant is basically refers to the allele which is always expressed in heterozygote. A wild-type fruit fly heterozygous for both genes was crossed with a fruit fly that was homozygous recessive for both genes. Table 7.1 is a summary of the cross. Complete table 7.1. So we can see wild-type parental phenotype. So obviously the parental phenotype will be red eye and long wing. Now the question says a wild-type fruit fly which is heterozygous for both genes. So heterozygous for both genes will be capital R, small r, and capital N, small n. Double homozygous recessive parents. Double homozygous recessive parents will have purple eyes and vestigial wing. And since it is double recessive, so it's going to be small r, small r, small n, small n. Okay. Now offspring phenotype. Now the offspring phenotype that we can see over here is red eye, long wings. We can see the next one, purple eye, vestigial wing. Then we can see red eye with vestigial wings. Now after that we can see purple eye with long wings. Now understanding offspring genotype. Red eye, long wings. So the offspring genotype should be, because it has red eye, so must have a red capital R, and long wings, so must have a capital N. Whereas the other parent is only homozygous recessive, so we'll have small r and small m from the other parent side. Because it has purple, the next one, because it has purple eyes, the purple eyes can only be possible if there is small r and small r, and vestigial wing is possible if there is small n and small n. Alright, the next one has red eye, that means there is one capital R with a small r, alright, from the other parent, and since it has short wings, so small n and small n. Alright, and finally purple eye refers to smaller and smaller, but long wings means there is one capital N, the other parent gives a small n. Explain why the four offspring phenotypes are not present in 1:1:1:1 ratio. Now the reason why we don't have 1:1 ratio is that two genes have autosomal linkage, so there is no random assortment. Large number of parental type offspring, recombinant, are only due to crossing over during meiosis. Guys, if you notice in the left hand side here, the explanation of how we got the offspring genotype is actually given over here. So as you can see from the diagram, one of the parent is double homozygous recessive parent with smaller and small n. So we only get small r and small n as in the genotype from the gamete, and whereas the other one is a heterozygous, so we write that down in the combination, and then basically we find out these organisms, and what we did is we matched it with their offspring genotype. Question number 8. In a continuous variation, a population shows range of phenotypes between two extremes with no distinct groups. Height and mass are examples of phenotypic traits that show continuous variation. Describe the genetic basis for continuous variation. Different alleles at a single gene locus have small effect on the phenotype. However, multiple genes can have an additive effect on the phenotype. Continuous variation is mainly caused by polygenes. Part B. Environmental factors can contribute to continuous variation. Suggest two environmental factors that may affect the body mass of an animal. So basically, the environmental factor can be such that the quantity of food that is available for the particular organism, the quality of food that is available for the particular organism, disease and temperature also can affect the body mass of a particular animal. Part C. Humans have used selective breeding, artificial selection for thousands of years to improve the quality of livestock. Outline the principles of selective breeding in livestock. So basically in selective breeding, humans apply the selection pressure. Humans can select parents that show desired characteristics, example, high milk yield. Then the parents are then bred together. The offspring with high milk yield is then chosen and then again bred with bull whose mother and grandmother had high milk yield. This process is then repeated over many generations to basically improve a particular livestock by artificial selection. Question number 9a. Describe how the structure of a mitochondrion is related to its function. The function of the mitochondria is to make ATP. Mitochondria has double membrane. The inner membrane is folded to increase the surface area. The mitochondria has ATP synthase molecule and electron transport chain in it. The inner membrane has site for oxidative phosphorylation and it is impermeable to hydrogen ions. The inner membrane has a high concentration of, the intermembrane space has a high concentration of protons. As protons are continuously pumped into the intermembrane space, this allows proton gradient to build up between the intermembrane space and matrix. The matrix contains coenzyme for link reaction. The outer membrane is permeable to pyruvate and oxygen. Mitochondria contains 70S ribosomes involved in protein synthesis. Question 9b. Explain how rice is adapted to grow with its root submerged in water. Rice plants have aerenchyma in stem and roots. Aerenchyma helps oxygen to diffuse to the roots. Shallow roots allows, it also assists the diffusion. Air film is trapped on under the leaves. This allows diffusion. When submerged in water, rice plants have greater internode growth, meaning they can grow larger as they are submerged in water. Growth is regulated by gibberellin. Anaerobic respiration can occur in roots when submerged in water. Rice plants is tolerant to high ethanol concentration. Rice plants can have alcohol dehydrogenase to break down the alcohol that they produce. And it can also tolerate very high ethanol concentration, which is a great advantage. Question number 10a. Explain how different types of gene mutation can affect the phenotype and outline the effects of the gene mutant alleles that cause Huntington disease on the phenotype of a person. Gene mutation due to base substitution often does not have a significant effect on phenotype. Base insertion or deletion leads to frame shift, so it has a significant effect on phenotype. Frame shift leads to changes in amino acid sequence. As a result, changes in the tertiary structure of the protein. Frame shift can also lead to premature stop codon. As a result, the protein made cannot function. Huntington's disease is caused by dominant allele. Huntington's disease has more repeats of base triplets CAG. Since HD is caused by dominant allele, heterozygotes will also have the disease because it is only needed one dominant allele to cause the disease. In HD, brain cells die more rapidly than normal. People suffering from HD experience mental deterioration. The greater the number of CAG repeats affects earlier onset of the disease. Question number 10b. Explain how gibberellin acts on DELA protein to stimulate the production of amylase in germinating seeds. DELA protein inhibit transcription factor PIF. When gibberellin binds to receptor in alluron layer, the gibberellin receptor complex binds to the enzyme that breaks down DELA protein. PIF then binds to promoter region of the DNA and causes the transcription of gene coding for amylase. Translation of mRNA then produces the amylase enzyme. Guys, that's all for today's question paper. So subscribe to our channel and basically in this way you're gonna get more and more question paper answers solved. All right, and basically recommend it to your friends if you find it useful. All right, and like the video, comment on the video if you want any related question paper to be solved. Thank you. Bye bye.